We will now discuss viral life cycle of coronaviruses. So let's start with viral binding and entry. We know that coronavirus spike proteins bind to a variety of cellular receptors depending on which virus and which group of coronaviruses are infecting the cells. Alpha coronaviruses bind to aminopeptidase N, which belongs to family of zinc-binding metalloproteinases. Murine hepatitis viruses can use adhesion molecules to bind and enter the cells, and we are interested in SARS. So SARS binds to metalloproteinase ACE2, which is angiotensin converting enzyme 2. It can also use lectin, L-sign, to enter cells. We also mentioned that beta coronaviruses, with exception of SARS, also express hemagglutinin esterase, which can bind to sialic acid. So once virus bind to the receptors, remember there is this S2 domain, which actually trigger fusion. So S protein, upon binding to a receptor, undergo conformational change, and S2 protein, which is a classical class 1 fusion protein, facilitate fusion of viral membrane with cell membrane. S protein can also trigger formation of syncytia, those multinucleated giant cells. Recall that S domain, because of those two, S1, S2 domain, can trigger exchange between different viruses that infect the same host, the same cells, and can expand the host range of the virus. Now, once a nucleocapsid and RNA is released into the cytoplasm, the replicase genes, meaning gene 1, genes which are in the gene 1, are translated as a polyproteins. And basically, there are two open reading frame. Open reading frame translate first, but it encounter ribosomes as pseudonode, which is between MP2 and polymerase, and this slowdowns or pause translation and allow frame shift. This frame shift allow translation of ORF1 afterwards. Now, genes 2 and 7, structural genes, are translated from subgenomic mRNA, but they are synthesized differently than those in toga viruses. And see here, we have N protein, M protein, E, and S proteins. So the replication, also the replication happens on the cytoplasmic membranes. Coronaviruses, like other viruses, trigger reorganization of the vesicle and membranes. And basically this complexes, this membrane structure induced by virus serve as a docking site where replication happens. Also, encapsulation of RNA occurs on these membranes. And if you look on this electron micrograph in first picture, you can see early membranes triggered by virus. And here on second one on the right, you can see cluster of those cell membrane which support virus replication. Now, genome replication proceeds via full-length negative strand intermediate. You recall plus RNA transcribed into minus RNA, and minus RNA afterwards serve as a template for synthesis of genomic RNA. Viral proteins are synthesized from subgenomic mRNA, and the subgenomic mRNA forms this cluster of mRNAs of different lengths, but it's the same 5' end. And we will come to it. How does it happen? So here it is. How does it happen? If you look on the 5', we have leaders and TRS sequence, which is on 3' end of leaders. TRS mean transcription regulating sequence. Now here is gene 1, and here are genes 2 and up to 7. each of the structural genes, 2, 3, 4, 5, 6, 7, on its 5' end have also TRS, transcription regulating sequences. So when RNA-dependent RNA polymerases attaches to 3' end of the genomic RNA and start to transcribe, it comes at the end of the respectively 5' end of the gene encoding structural protein comes to this TRS and it slows it down, and often it falls off the template RNA. And when it does disconnect from that RNA, it is targeted to this TRS, which is on 3' and off leader. So because they are complementary sequences, they anneal, and polymerase completes the transcription. So all this minus and subgenomic RNA will have the same 3' end, okay? Now, like this, so one transcription end after gene 7, and other end after gene 6, after gene 5, and so on, so on. So this creates a set of minus and minus strand subgenomic mRNA. And this minus strand subgenomic RNA are afterwards transcribed into plus and subgenomic RNA. And those, because they are transcribed based on this subgenomic mRNA, have the same 5' end, even so they have different lengths. So TRS, transcriptional regulating sequences, are critical for synthesizing this set of subgenomic mRNA with minus strand, and those are afterwards transcribed into plus subgenomic mRNA, and those serve as a mRNA for synthesis of structural proteins, and some of them encode several, like they are polyproteins, which are afterwards cleaved. So this discontinuous model actually, beside or together with a recombination of spike protein, can explain recombination between different viral genome. Why it is? Because when this minus RNA falls off and another synthesis is initiated, if there are two genomic RNA, let's say two different coronaviruses infected the same host, the same cell, basically that RNA polymerase can jump, can switch and use that secondary genomic RNA from different virus as a template, which would create genomic mix of minus subgenomic RNAs, and therefore mix of genomic, respectively subgenomic plus mRNA. So the switching of RNA polymerase between two different positive strand RNA, if they are in the same cell, can contribute to recombination and creation of new coronaviruses, which can have different tropism, different characteristic, and different pathogenesis. Now, once the RNA is synthesized, genomic RNA is synthesized, we know that those spike proteins are targeted to ER membrane, and M protein help to bring nucleocapsid. So it's very important for assembly. So virus start to assemble and form immature virions inside of ER and Golgi. And as it travels through Golgi, those polyproteins are cleaved, and in those secondary vesicle, virus is brought outside of the cells. So basically, virus is released within those secretory vesicles. Now, we talk about S protein. Stop and restart synthesis of subgenomic RNA, which can help to create changes. And these high changes, together with error in RNA polymerase synthesis, it does not really have proofreading capabilities. So this lack of proofreading, together with this RNA-RNA recombination, spike recombination, can actually trigger evolution and synthesis of new viruses, new coronaviruses, and those can trigger epidemics and pandemics because humans and animals do not have any immunity against those new recombined viruses.